Hello all. In this video, I'm going to show how to use Jalview for multiple sequence alignment and analysis. So let's start. So in order to download Jalview, so first we will have to visit uh, Jalview website. So you have to type Jalview in the Google search bar, and then this is the result. This is the top result. You have to click on what is Jalview, and this green arrow is showing that uh, it's related to download. So you have to click here, and then this is the installation file. This is the so when you will click on this, it will, so the executable file will, so in order to download, so I have already downloaded this software, so I'm going to cancel it. So executable file is here. So in order to install Jalview, you have to right click on this executable file and click on open, run. So I have already Jalview on my laptop. That's why it's showing that update the existing installation. So I don't want to update it, and I don't want to install into a different directory. So if you are installing Jalview first time, then this option will be not available. Only it will show where the Jalview will be installed. So you can change the directory, but default location will be in C drive. So I'm going to cancel this setup. So let's so Jalview has been already downloaded here. So once it's downloaded, then you can open it either from here. So in search, you can type Jalview and click on Jalview, or either on desktop you will see the Jalview icon and you can click on that. So these are the example, actually the all examples files have been uploaded here. So you need internet connection for Jalview software. So I'm going to close this all windows except the main window. So this is the main window and you don't have to close it. So now for multiple sequence alignment, we need multiple sequences. So from where we will get multiple sequences? So here I'm going to use UniProt. So UniProt is a database of protein sequences and it's a freely available. So you have to click on UniProt knowledge base. So this is UniProt KB, UniProt knowledge base, and it has two parts, SwissProt and Tremble. So SwissProt has manually annotated and reviewed protein sequences, and Tremble is translated in MBL, and it has automatically annotated and not reviewed sequences. See, so in order to search sequences, just type the name of the protein, type name of any protein. So I'm going to search myoglobin. So here you will, so when you will press click on search, then you will get the results related to myoglobin. So here are 1782 results related to myoglobin, and you can increase this number like in. So let's in one page I want to show 100 sequences, so it will update the page, and I'm going to select here 10 sequences, and they will be from different organisms. So first is mouse, second is from human, third is from rat, fourth is from bovine, fifth is from zebrafish, sixth is from chicken, seventh is from pig, eighth is from pig roundworm, ninth is from chimpanzee, and 10th is from Indian elephant. So all 10 sequences have been selected. So I'm going to download all these sequences. So for this, you have to click on download. And here it is showing that 10 sequences have been selected. And if you want to download all, then you can download all sequences. But here only 10 sequences. We need only 10 sequences. And it is also showing that whether you need the file in the faster format, or you can choose here also different options. And I'm going to use this uncompressed option and click on go. So all 10 sequences here, so you see that all 10 sequences are appear on the screen, and you can select all 10 sequences and then copy, click on copy. Then I'm going to paste all these 10 sequences in a notepad file. So paste here, and then click on save as. So myoglobin.faster. So I have already this file, that's why it will ask me to overwrite the file. So save. So do you want to replace it? I don't want to replace it, so yes. So I want to replace it, so yes. So now this myoglobin.faster file is located in mmfvvic2 folder. So here myoglobin.faster, and now we will use these all 10 sequences to perform multiple sequence alignment, and we are going to use Jalview, which is a freely available software. So in order to perform multiple sequence alignment, first you need to load all 10 sequences. So go to file, input alignment, and from file. So you have to first open the file which you have created. So if you want to open any file containing sequences, then you have to locate that file. So here I have created myoglobin.faster. So I'm going to open myoglobin.faster. So this will a new window will appear, and it has all sequences, and you see that these are entries, and SP stands for Swiss prot. This is Uniprot ID, is the organism name, and this is the range, residue range. Okay, so in order to perform alignment, we need to click on select, then select all, then go to web service, click on alignment, and here I'm going to use muscle bit default. So you can change the setting also. So I'm going to use the default setting. So it will take, in this case, it will take less than 1 minute. So now the alignment is over. So you can see these are all aligned sequences. So, but this alignment is not attractive. So, in order to make the alignment attractive and more informative, we need to color them. So, in Jalview you have different coloring options like cluster X. You can color according to the Blossom 62 score, according to percentage identity, hydrophobicity. You can also define your own color. So here, the first option, I'm going to use the cluster X. So cluster X coloring is based on physiochemical property of residue. So like if it's a hydrophobic, then it will be colored differently. If it's a charged, then it will be colored differently. So here you see that the K and also lysine is a positively charged residue. Then it's color, it has red color, and then like the background is red. It's not. And then we have also let's search here. Arginine is also positively charged, so it has the background is red. If you want to color it differently according to the percentage identity, then you see that here you can change it's according to the percentage identity, and then you can also uh, choose the threshold. So suppose you want to color all the residues above 90% identity, then all those residues which are showing 90% greater than 90 or 90% identity, they will be colored. So in case of 100%, you see only those columns, so these are the columns, only those columns will be highlighted which will have 100% conservation. You can change this threshold, so you can change it to 95%, 90%. Okay. And in order to find a motif, you can go to select and find. So suppose you want to find a conserved motif, and suppose it's a DFG. When DFG, so you will be able to find it. So DFG is here. So you can see here the DFG is not fully conserved in all on all 10 sequences. So here you will see there are different rows, and I'm going to delete two rows here, conservation and quality. So this is not important. These are not important. So I'm going to retain one. I'm going to show only how this consensus row and occupancy row. So in consensus row, when you will right click, then you see this option show logo. So it will show beautiful logo, and you can like by when you will place the left button of your mouse, and then uh, you hold the button and just try to uh, drag it. So you have to click on this and then you try to drag it, so you can uh, make it more clear. And you see that based on consensus sequence, like this is based on consensus sequence. So basically it is according to the frequency of the residue in particular in particular column. So in this column we see that V is like it has like it is most prevalent residue. So that's why the V is here, it has been highlighted. Here it is L. The leucine is also um, it's it is most prevalent. So it's a L is 90%. So the residue which has highest frequency will will be highlighted, and it will have like the size of the later will be more. Like, and if you have like here, we see that we need to scroll it. So in order to see whole sequence, we need to scroll it. So if you don't want, then you can click on wrap. So wrap will actually try to fit all the complete alignment in single window. So you so scrolling is not required here, and you can export this alignment either as an image, or you can save it uh, as sequence file. So you can save it as aligned dot faster, myoglobin aligned dot faster. So I'm going to overwrite it, and if you want to save it, you want to export the image, then just click on PNG and just give a name, myoglobin aligned dot PNG, and then it's okay. And you can also decrease the size of uh, you can also decrease the size of these letters. So you can set the font size. So basically this single letter code of residue amino acid is you can just decrease the size. So suppose you want to set it at you want to make it 8. So still you see that this is visible. So you see that alignment has gaps also. So these gaps are because because this length is different from this the wrap. So if you have two, suppose you have two sequences and they are not equal in length, then definitely there will be gap because the program try to optimize the alignment. So so in that process it will create gaps. So you see that here this zebrafish, the myoglobin of zebrafish is 147 amino acid long and residue long, and while the mouse, human, rat, even chimpanzee and elephant, pig, they have 154 amino acids. So that's why you see gaps here. So I think it's now easy to perform alignment, and you can export beautiful picture of your multiple sequence alignment, and you can also perform pairwise alignment. So suppose if you want to compare, so you can do many things, like if you want to compare only human and elephant sequences, then uh, press the control key and hold it and then select multiple sequences. So when you will click on control key and hold it, you can select multiple sequences and then go to calculate and pairwise alignment, and it will perform pairwise alignment. So myoglobin of human has 84.42% identity with human myoglobin. So this dots are showing that these residues are not same, are not identical. This vertical line is showing that this residue is identical. You can also create, you can extract scores, you can, so if you want to sort these sequences by pairwise identity, uh, then this rearrangement of these sequences will occur. So you see that this is now the alignment is according to the pairwise identity. So in this way, you can you can perform multiple sequence alignment of any protein, and you can analyze. So Jalview has many options, and you can explore all options. See this alignment, secondary structure prediction, protein disorder analysis, conservation, phase TV differences. So if you want to bring the scale above, then the scale will appear. So now I'm going to end this session. So hope you will be able to enjoy the multiple sequence alignment using Jalview. And yes, you can analyze. So I'm going to make another video in which I will uh, I will do structure visualization of myoglobin protein, and I will use uh, this sequence alignment to locate those residues which are conserved. So you see that there are many residues which are highly conserved, like 100% conservation. So we will see that why we will try to locate this, we will try to map these conserved residues on 3D structure of myoglobin, and we will try to analyze that why these residues are conserved. So okay, so thank you for watching this video, and don't forget to subscribe the channel. Thank you very much.